국제백신연구소 IVI는 전 세계 개발도상국, 특히 어린이 전염병 예방 백신 개발을 위한 세계 유일의 국제기구입니다. IVI는 안전하고 효과적이며 저렴한 백신의 발굴, 개발 및 보급을 사명으로 세계 전역의 협력기관들과 함께 필수적인 백신의 개발을 촉진함으로써 전 세계 가난한 사람들이 건강하고 생산적인 삶을 향유할 수 있도록 돕고 있습니다. 현재 IVI는 콜레라, 장티푸스, 이질, 살모넬라 등 연구 프로젝트를 아시아, 아프리카, 남미의 30여 개 국가에서 실시하고 있습니다. 질병으로 고통받는 사람들에게 살아갈 권리를 찾아주고 지구촌 아이들에게 생명과 희망을 선사합니다. 인류를 위한 IVI의 백신 개발은 대한민국과 스웨덴, 인도 정부, 빌 앤 멜린다 게이츠 재단, 웰컴 트러스트 등 여러 자선기관, 기업과 개인의 후원으로 시행됩니다. 우리 모두를 위협하는 신종 감염병. IVI는 코로나19 등 신종 감염병 백신의 개발에도 적극 참여하고 있습니다. 국경이 없는 바이러스에 대응에는 국경이 없는 백신 연대가 필요합니다. 전염병으로부터 해방된 세상이라는 우리의 비전을 공유하는 모든 이들이 백신의 공평한 보급을 위한 IVI의 노력에 함께해 주시기를 당부드립니다.
Good day, and thank you for the opportunity to speak today for the World Science Culture Forum on the topic COVID-19 vaccines. How much do we know? The global response to COVID-19 has been dramatic, and some countries have been more successful than others at controlling the extent of the pandemic in their populations. What you see on the graph on the right are the impact of different countries' efforts in controlling COVID-19 in the absence of a vaccine. So you see at the top of the graph, the United States, India, and Brazil, which have been having particular problems in controlling the spread of COVID-19 within the population, and then other countries such as South Korea, China, Canada, France, which have done a relatively better job at controlling the outbreak of COVID-19. The mechanisms to control have been the same in many countries: testing, tracking, isolating, treating, distancing, masks, hygiene, public support. In many countries, not necessarily in Korea, they've had to institute lockdowns. That is a complete suspension or a partial suspension of all social activity, including economic activity. And these have had very dramatic consequences around the world. But as of September 26th, we had almost 1 million deaths from COVID-19 and over 30 million infections, and the number continues to increase. What is normal now? And what you see in the graph are the impact of the different waves. In the United States, there never seemed to be the end of the first wave. The same is true in India, with an initial small suppression during the lockdown with a resurgence of cases thereafter. With France, an initial suppression and success, and then followed by a resurgence over the summer. The same is true, but to a lesser extent, in Italy, which had actually some of the highest levels of COVID-19 infection in the spring. South Korea has been relatively spared. The initial round of virus spread in February and March was brought largely under control, with then successive outbreaks, small outbreaks, the one in Itaewon, the one associated with the Presbyterian churches in August. But again, these are slowly coming back under control. So the new normal is a series of either complete, partial, or incomplete suppression of virus spread, punctuated by outbreaks, sometimes substantial outbreaks. You know, in the United States, it could be college students. It could be particular locations. It could be political rallies. So again, you know, the ability of the governments to maintain the new normal with the kinds of controls that are necessary in order to prevent spread in the absence of a vaccine are going to characterize the more successful countries and will fail in those countries that have been less successful. As you're developing a new vaccine, several issues come up commonly. The first is, does infection provide immunity against reinfection? And you've heard this raised time and time again with regard to COVID-19 vaccines. Why is that? In the classic vaccine disease model, such as hepatitis A, although with hepatitis A infection you can have asymptomatic infection or people can become very jaundiced and turn bright yellow, by and large the body's protective or immune response will initially control and eventually eliminate the virus from the body. And those same protective responses will protect the body against reinfection. For some viruses, such as HIV, that level of control is never achieved. So with HIV infection, the body is never able to clear infection. The immune system cannot defeat the virus on its own, and people will progress to AIDS and death if left untreated. What we also know is that same protective response that fails to control HIV is unable to prevent reinfection. So a person can actually be what we call super-infected with HIV even though they have an ongoing HIV infection. We don't think that that's the case with COVID-19. With COVID-19, there are already 3 monkey studies that show that if a monkey's been infected with COVID-19, it is immune to reinfection, at least for a certain period of time. We think that the same is true in the setting of natural infection of humans, although again, it's been difficult to actually make that, to prove that hypothesis. A second issue that's come up actually more recently is the question of whether or not we'll need seasonal SARS-CoV-2 or COVID-19 vaccines. And it gets to the question around the amount of sequence variation there is in COVID-19. The best way to think about this is that COVID-19, the amount of sequence variation in the 70,000 or so full-length sequences that we've done so far is about the width of the tip of a pin. So if you made a pin prick in a card, that would be the amount of sequence variation in COVID-19. If you were to look at influenza infection in a single year, you'd see an amount of sequence variation about equal to the width of the tip of my finger. If you looked at HIV in a single year, the amount of variation is the size of my palm. So COVID-19 actually has a very small amount of sequence variation. So in answer to the question, will COVID-19 be like influenza, which requires seasonal vaccines every year, or rotavirus, where a single vaccine made before 2007 still protects today, the evidence would tend to support a single vaccine. Again, though, we've only known about COVID-19 for 9 months. And whether things will change in the next year, 2 years, or 3 years, we really can't tell. The bottom line is, though, that compared to influenza, COVID-19 has 10 times less sequence variation. The next issue to come up is, so we know that people who have been infected with COVID may be protected against reinfection, but what is it about the immune or protective responses in those individuals that actually protects them against infection? Is it infection-fighting protein, what we call neutralizing antibody, proteins that bind to and inactivate the virus? Or is it killer T cells, these cells that kill virally infected cells? Or is it a mixture of both? And it's a mixture of both, with antibody predominating, so that the neutralizing antibodies are often found to be what we call the correlate of protection. But in fact, with COVID-19, we, at this point, don't know. Another issue that comes up quickly when you're developing a new vaccine is there an animal model? Why is an animal model important? Well, we can't get information on protection of humans with a vaccine right away. That takes a period of months, in the usual cases, 5 to 10 years. But we can test if we can infect an animal with the virus. We can then first vaccinate the animal, so in this case a non-human primate, a monkey, and then challenge them with COVID-19. And if a monkey is protected by vaccination, then it's possible that the humans will also. With COVID-19, we're actually a bit lucky because you can infect certain mouse species that are genetically modified. You can infect hamsters, you can infect ferrets, and you can infect monkeys. And in all of those circumstances, vaccinating them first results in protection. Now, will that mean that there's a 100% likelihood that the COVID vaccine will be successful? No, it doesn't. But what it does mean is that at least in animals, we are able with a vaccine to develop the right protective responses that protect the animal against infection and or disease. So far, 12 different vaccines have shown protection against COVID-19. How that will translate into human data, I think we're still waiting to hear the answer to, and hopefully by the end of this year, by the end of 2020, we will start to see evidence of whether these vaccines work or don't work. The final issue that arises happens to be around safety. And everyone knows that we give vaccines not to people who are sick, we give vaccines to people who are healthy in order to keep them healthy. So that vaccines have a very high standard that they have to pass. Vaccines have to be safe. They not only have to be effective, but they have to be safe. And in particular with SARS-CoV-2, there are two questions that people have asked, not based on SARS vaccine research, SARS-CoV vaccine research, but actually on the original SARS virus that came in 2002. When we were trying to develop a vaccine against that 2002 SARS-CoV, we found that in certain mice species, in certain mouse species that received vaccine and then were challenged with the virus, that the vaccine protected the mice against infection, but the mice developed a second kind of disease in their lungs, kind of like an allergic disease, and that was seen occasionally when they tested the same vaccine in ferrets, and in one occasion in monkeys. When we looked with the SARS coronavirus, which is a cousin of the MERS coronavirus, which is a cousin of SARS, we didn't see it as frequently. With SARS-CoV-2, we've now done 12 monkey studies. We've vaccinated the monkeys, we've challenged them with COVID-19, and all of the monkeys were protected, and none of them have developed this new disease. Does that mean that we can ignore it? No, it doesn't. It means that we have to commit to following human volunteers in clinical trials for a longer period of time to be able to ensure that they are safe and to assure the public that any vaccine developed for COVID-19 is both effective and safe. Under normal circumstances, it takes 5 to 10 years to develop a vaccine. The cost for a big vaccine company like Merck or GSK is between $500 million and $1.5 billion, and the failure rate from laboratory to licensure is 93%. That means that if you start here in the discovery phase, inventing a new vaccine, you test it in animals, you take it through human clinical trials, phase 1, phase 2, and phase 3, only 1 out of the 10 vaccines that you started with in the laboratory will actually end up being sold as a successful vaccine. So how then are we going to proceed to develop a COVID-19 vaccine in 6 to 18 months? And that's going to be a very difficult task, but one that many governments around the world and organizations around the world, including IVI, have committed to. What you see in this slide is, on the top panel, an example of a vaccine that would take 5 years to develop. So we do the work in animals, what we call the preclinical work. Then we move to the first stage in humans, called phase 1, typically with 50 or fewer volunteers. Phase 1 is really designed to look at safety. Is the vaccine safe? Are there any bad side effects that we see right away? We can also collect a small amount of information on the protective responses that are generated by the vaccine. Is it making infection-fighting protein? Is it making the killer cells? But primarily in phase 1, we're focused on safety. If it meets those criteria, we can proceed to phase 2, which typically lasts a year. And in phase 2, we are concerned about the target population, the population of people for whom the vaccine is designated. So if it were for the elderly, we test it in an elderly population. If it's for babies, we would have to test the vaccine in babies. In phase 2, which involves hundreds of volunteers, we are really interested in the correct protective responses. Infection-fighting proteins, neutralizing antibody, for instance, or killer T cells. Are these being generated by the vaccine in the target population? And if we're satisfied with that answer, we can proceed to phase 3. Now, phase 3 has thousands, sometimes tens of thousands of volunteers. So you can imagine they're complex and they're very expensive. So a company, before it moves to phase 3, wants to make sure that everything is optimized. Everything that they could do to make this vaccine successful, they've done. And that tends to drive the timeline of 5 to 10 years and a significant proportion of the cost of the $500 million to $1 billion of developing a vaccine. If in phase 3 the vaccine is safe and shows protection against infection or disease, then the company presents all the information to what we call a national regulatory authority. In Korea, that's the Ministry of Food and Drug Safety. In the United States, it's the Food and Drug Administration. They typically take 9 to 12 months to review all of the data. And in the old days when we had paper, it was hundreds of thousands of pieces of paper. They would sometimes completely redo the statistical analysis to prove to themselves that the vaccine, in fact, protected against infection and disease and was safe. Now, we won't have that luxury in COVID-19. And so what's shown now on the bottom panel is what we anticipate will be the course of development of a COVID-19 vaccine within 12 months. So the preclinical phase now, the animal testing phase, will overlap with phase 1 testing in humans. Now, how can you do that? You do it with vaccines that have a track record of being safe and effective in humans. So, for instance, DNA vaccines have been in thousands of volunteers. RNA vaccines have been in thousands of volunteers. So we can safely begin the animal testing and begin the human testing at the same time. Typically, we give 2 doses of the COVID-19 vaccines. So 2 weeks after the second dose of COVID-19 in phase 1, we can look at the safety data and say, okay, there weren't any significant side effects. Let's proceed with phase 2. Phase 1 will still continue for the rest of the year. We'll continue to follow up volunteers for safety. But phase 2 can be initiated on the basis of demonstration of safety in phase 1. So we've overlapped phase 1 and phase 2, telescoped them. Phase 2, again, will have 2 doses, and several weeks after the final dose is administered in the final volunteer in phase 2, we can start to analyze safety and whether or not protective immune responses are developed. And that may take a few months, but at the end of the day, if we're satisfied with safety and immunogenicity, that the vaccine is making the correct protective responses, then we can begin the enrollment of phase 3. So you can see now that 8 companies are currently in phase 3 trials around the world, and those companies followed a pathway very similar to this one, where they telescoped phase 1, phase 2, and phase 3 in order to accelerate the testing of a vaccine. Now, depending on the number of infections, a vaccine you can actually look at the results in a vaccine trial. Typically, these are what we call pre-specified analyses. The company just doesn't go in and say, I think I want to look now. These are analyses, these are statistical analyses that the company agrees to with the national regulatory authority. So the Ministry of Food and Drug Safety or the Food and Drug Administration in the United States has said, we agree with you, you should look at the data at this point and at this point and at this point, and we agree, you should do that. And the company commits to doing it, and when they look, if they find that the vaccine is safe and effective, they may move forward and request an emergency use authorization. Or they could apply for licensure. More likely to be emergency use early on because the data will not be complete. Phase 3 is still going on. Emergency use allows a company to then make the vaccine available within the country. Still on an experimental basis, people still have to be told that the vaccine hasn't been formally approved. People still have to be followed up for efficacy and safety. But it gives more people access to the vaccine earlier. And that's how you would likely do it. Now remember that the signal for efficacy, whether the vaccine protects against infection or disease, will be the same in the 5-year model or in the 12-month model. The difference that we see is the difference in the long-term follow-up for safety. So if you have 5 years or if you have 10 years, you've been able to look at the vaccine safety profile for a longer period of time. We will probably have 6 to 9 months worth of safety information. The only good news in that is that most of the side effects occur in the first 2 weeks after vaccination, and the vast majority occur in the first 2 months, and we should have that information. But there still are examples of vaccines, for instance, a vaccine against dengue, where the late-term side effects, the side effects weren't visible until year 2 or year 3. And those are the things that we need to commit to following up in the people who've courageously volunteered to take vaccines in phase 1, phase 2, and phase 3. So, how do we get from a timeline of 5 to 10 years, a cost of $1.5 billion, and a failure rate of 93%? How are we going to convince companies that it's in their interest to accelerate a timeline that normally is 5 to 10 years and reduce it to 6 to 18 months? Well, the US government approach to accelerate, they need to de-risk the trial for the companies. In order to de-risk, they had to fund the companies. So if you look at what the United States government has done, it's provided $10.3 billion to the companies listed there. In addition, the companies have committed to the production of hundreds of millions of doses of vaccine. So you can see AstraZeneca 300 million doses, Johnson & Johnson 100 million doses, with an option to purchase an additional 200 million doses. And you can see all the spending down there, all the way down to emergent, ology, and SiO2 materials science, which are contract manufacturers. So in case the US government has to manufacture additional vaccine or wants to manufacture certain of these vaccines in advance, they can by using these contract manufacturers that have the capacity to manufacture tens of millions of doses of vaccine. In addition to US government funding, the Coalition for Epidemic Preparedness Innovations, which Korea has joined, was able to provide a significant amount of funding to some of the companies early on. So for instance, the Oxford Group, which eventually merged their effort with AstraZeneca, Moderna, the RNA vaccine, Inovio, the DNA vaccine, all received funding from CEPI. And the good thing about CEPI is that when you take funding from CEPI, you commit to global access and to providing the vaccine at a reasonable cost around the world. Now, it's difficult to talk about the 200 or so candidates that are in the development phase for COVID-19 vaccines. So it's easiest to focus on the 43 that are currently in human clinical testing, and particularly on the 8 that are in phase 3, so the test of efficacy and safety. This is just a chart from the World Health Organization website. This is actually the most recent data that's available, 25th of September, and then from the Wikipedia website, which is actually updated continuously and is also a good source if you're interested in looking at exactly what vaccines are out there and what stage of development they're in. This slide actually is more helpful because it goes through the major candidates in the COVID-19 vaccine race. What it shows you in the first circle, for instance, for Moderna, it's in March, is when the first volunteer was given a vaccine. The second circle is when the first data from the human clinical trial became available. The third circle is when the late stage, the phase 3 or phase 2/3 study started. And the hatched bar is where production of the vaccine will start. So you can see that for Moderna, for Pfizer, for University of Oxford AstraZeneca, for Johnson & Johnson, GlaxoSmithKline, and Sinopharm, which is a Chinese company, production has already or will soon start of vaccines that have not yet been shown to be safe and effective. Why are they starting early? Because countries and companies have committed to purchase doses in advance, and I'll have a slide on that in a little bit. What you see with the yellow stars are companies that have already received emergency use approval. So for instance, you could actually receive the Sinopharm vaccine in the United Arab Emirates, which has also issued an emergency use if you're a healthcare worker. If you are a healthcare worker in China or a member of the government or working for the company, Sinopharm, hundreds of thousands of doses have been issued. So what evidence do we have for these vaccines? What I've shown here are many of the vaccines, or actually 12 of the vaccines, for which non-human primates, so monkey challenge data are available. So these are monkeys that have been vaccinated with the vaccines shown on the left, and then were challenged with COVID-19 to see if the vaccines will protect against an infection and disease. And you can see that in all but one of the cases, we make what are called neutralizing antibodies, proteins that bind to and inactivate the virus. And in all of those situations, except for CanSino, which has not reported out its results yet, there has been protection against COVID-19 infection, and there has been no evidence of that late infiltrative pulmonary disease that we were worried about in safety. So far the monkey studies have been reassuring in that the vaccines, all of them, have appeared to protect against infection and disease. And a number of those candidates have now moved on into phase 3 testing, and those are shown on the far right-hand side. What I've shown here are the human clinical trials of vaccines that are in phase 3 or soon to be in phase 3. And what I've done is I've divided them into categories. So what you see in the gray box are the vaccines whose neutralizing antibody titers, the level of neutralizing antibody, is less than 100. What you see in the next box is the group of vaccines whose neutralizing antibody levels are over 100 but less than 500. And what you see in the green are vaccines which have a very, very high level of neutralizing antibody. And so you can see that all of them make neutralizing antibody, but at different levels. Now, do we know that neutralizing antibody levels correlate with protection? No, we don't. We suspect that they do. Could other antibody be important? Yes, in fact, there are some data from monkey studies that would suggest that not only neutralizing antibody, but other types of antibody that help the body to clean itself of virus or viral particles may also be important. But until we have data from a human phase 3 trial, all of this will be speculation. One of the caveats is, although I've been able to segregate the vaccines by neutralizing antibody titer, there is no standardization of that test. So there's no guarantee that a test done by CanSino or a test done by Johnson & Johnson will give you the same result if they were used on the same sample. So as we move to compare vaccines, we are going to have to compare the vaccine-induced protective or immune responses in a single system with a single group of people doing a standardized assay, and that's going to be very important. Otherwise, we'll again be guessing. So proving that a vaccine works, and I think that by the end of this year or early next year, we may have proof that a vaccine works or proof that it doesn't work. But the next big question is, can we make it? Can we make that vaccine? So CEPI has gone around the world and has found that around the world, we have the capacity to manufacture 10 billion doses of vaccine. Now, if you think that everybody on the planet needs to be vaccinated against COVID-19, there are 8 billion people. If they need 2 doses, that's 16 billion doses of vaccine. So the 10 billion dose maximum doesn't necessarily take into account different types of vaccine. So not all factories can make all types of vaccines. Sometimes it's very specific for whole inactivated vaccines or very specific for adenovirus vaccines. So again, we're going to need to look at that more carefully. An organization called COVAX, which is actually a group of 170 countries being led by the World Health Organization, Gavi, the Vaccine Alliance, and CEPI, the Coalition for Epidemic Preparedness Innovations, have committed to identifying and procuring 2 billion doses of World Health Organization approved vaccine by the end of 2021. And that would be a great thing, and I'll have another slide on COVAX subsequently. Operation Warp Speed, which is the US government program, has also, as you saw on a previous slide, contracted for hundreds of millions of doses of vaccines in anticipation of the vaccines actually being shown to be safe and effective. And finally, companies like AstraZeneca, like Moderna or Sinovac have been going around the world to identify other vaccine manufacturers who can take the vaccine from Moderna and manufacture it in bulk and help the company to distribute the vaccine around the world. So hopefully, with this additional contract manufacturing and licensing of vaccines, with the capacities that have been identified by the US government through Operation Warp Speed and by CEPI, we will be able to get a certain number, hopefully 2 billion doses, by the end of 2021. But I think the important thing to recognize is that in the end, we should not think of this as a US vaccine or a Belgian vaccine or a Russian vaccine or a Chinese vaccine. This is a race that humanity has to win, not a country. And really, do the congratulations, do the laurels go to the first company that proves that a vaccine will protect, or does it go to the company that can provide the hundreds of millions of doses that the world needs in order to stop the COVID-19 pandemic? Now, when we talk about the world stopping the COVID-19 pandemic, we have to talk about COVAX. Now, this is a collaboration. It's being led by CEPI, the World Health Organization, and Gavi. 174 countries have signed on to COVAX. They're still trying to negotiate the very complex deals that will allow 2 billion doses of WHO pre-qualified vaccine to be given out around the world. This will account for roughly 20% of a country's anticipated need for vaccine. That will allow all the countries participating in COVAX, if they're successful, to vaccinate the elderly and the health care workers, 2 very critical populations, and populations that need to be protected early on as soon as we have a safe and effective vaccine. The operating principles of COVAX: global access, impact orientation, transparency, solidarity, and collective ownership of the solution to the COVID-19 pandemic. Well, we do need equity and access with COVAX, but when you look at the current situation, and what you see on the left-hand side of the Sankey diagram are the companies, AstraZeneca, Pfizer, Moderna, et cetera, and where the doses have been pre-purchased. So the wealthy countries, the high-income countries, have pre-ordered the bulk of vaccine that currently is on future consignment. 1.6 billion doses for the US, 1.5 billion doses for the European Union, 400 million doses for the United Kingdom, 300 million for Japan. You don't see a middle income or low income country among them. And for them, COVAX will be critically important in order to avoid what we call the implementation gap. And I'm going to use as the example for this a vaccine that actually is a great vaccine and is being distributed all around the world. But the rotavirus vaccine, which is a vaccine we give to children to prevent diarrhea, is being used everywhere. It's being used in Korea, it's being used in the United States, and where it's been used, it's managed to dramatically reduce the impact of rotavirus diarrhea. So you don't find children with rotavirus diarrhea in Korea or in the United States because of the effectiveness of the vaccine. That vaccine was approved in the United States in 2007, and by 2014, 70% of American children got 3 doses of rotavirus vaccine. It was approved by the World Health Organization in 2009 with a recommendation for use, but by 2014, less than 20% of children around the world received that vaccine, received all 3 doses, and by 2020, less than 40% of children receive all 3 doses. That kind of gap, the implementation gap, cannot be allowed to exist for COVID-19 because that will simply prolong the epidemic and make more difficult our ability to respond to it. So final thoughts. We'll likely have evidence of efficacy and safety later this year or by early in 2021. But what does the day after efficacy look like? The day after someone stands up with much celebration and says, we have a vaccine that is safe and effective. Well, first, who's going to make it? Second, how are we going to ensure that equity and access are provided to low and middle income countries and to high income countries around the world that can't necessarily spend billions of dollars in order to pre-order vaccine? And then there are a number of other really significant questions that are scientific questions, but that must be answered in order to make sure that we have the best possible solution from a vaccine perspective. First is, have we been able to optimize the dose of vaccine, the schedule of vaccine? Will we have to give booster doses of vaccine? All these things are unknown and have to be studied the day after efficacy. We should determine something called a correlate of protection, a laboratory assay that predicts whether a vaccine will work. Now, why is that important? Well, once you have that laboratory assay, that correlate of protection, you will greatly simplify clinical trials. You will no longer need 40,000 people in order to prove that a vaccine should work. You should be able to do it with hundreds, if not a few thousand people. And that will make it faster and cheaper to develop new and better vaccines for the future. Finally, we need something called real-world evidence. We need to do what are called effectiveness trials. So it's one thing for a vaccine to work in a clinical trial where 15,000 people get vaccine, 15,000 people get placebo, and then we compare the number of infections in one versus the other. That's a very controlled situation. What happens when we get out into the real world and when we have to deliver vaccine by donkey, or where we have to ship a vaccine that has to be kept at minus 70 degrees to parts of the world where we can't guarantee that a vaccine is stored at 2 degrees? How are we going to show the real-world efficacy, effectiveness of the vaccine? And that gets to actually a more fundamental question around herd immunity. Everyone's heard us talk about herd immunity. You know, we could let the disease run through populations, and when 70% or so of the population is infected, when millions of people have died, we would be at herd immunity. So the virus would no longer efficiently be transmitted in a population. But vaccines can do that by imitating infection. So if we, theoretically, were to vaccinate 70% of a population, if that vaccine were suitable, we might see herd immunity. But in order to make that, in order to say that, we have to prove it. And so the trials to evaluate herd immunity need to be designed now, need to be funded when the vaccines are available. And we really need to know, are we okay just vaccinating 70% of the population? Or will we have to achieve much higher levels of vaccination in order to see an end to the COVID-19 pandemic? Additional questions around long-term safety and licensure versus emergency use are things that we will have to wrestle with the day after efficacy. And these are problems that will plague every vaccine as their data become available and as they're shown to be safe and effective. You know, hopefully we'll have more than one. But that complicates matters also, because if you get one vaccine, can your booster dose be a different vaccine? And again, we have no idea. So how long and hard will it be to get back to the old normal? That's the question. It won't be fast. Hopefully with the right vaccine solutions, we can make the process of transitioning back to normal less painful. Thank you.